to get electricity in the university, to get a microscope, how much the struggle we had to find. This is say that this is something like winning a Nobel Prize in the University of Mysore with from zero. No, but he has appreciated, he wrote on behalf of this in several blogs in the Facebook there. He has convinced many people that this kind of work has been done also. Yes. So he is propagating this is the kind of the good work what they are doing in the university. So what he said that other faculty in the IAC not comparable to the university. Sometimes the university faculty do better than them also. That is the way. So we have to compete, not curve. So okay, this is the one I said. This is the reason undergo recombinations. Because protocol gene was there, we not be identical. These are the duplication we should be able to record that we repeated, we got this also. So far, no one said that this is what we did it wrong because we were very careful when we do this and the analysis. These are several about 10, more than 10, 15 papers came out with this kind of copy number variation. Somebody asked about Parkinson. Some of the students asked about non-distinctions. A different kind of interest studied. We have about 44,000 CNV. Where exactly mapped? I mean, we made that kind of analysis also. Even dyslexia, autism, they identified certain variants and we published them. So last part here is that we have genome, we know genomic variation, how genomes have been evolved from bacteria to the eukaryotic higher order of the system. Few glimpses we can record it here. As already told you, eukaryotic system have lot of duplication, transposition, and mutation, and also in polymerization. They are not diploid. Some of them are tetraploid, the plants. So first transitions of the first eukaryotic system, that is 1.4 billion years ago, have 5,000 genes. I'm talking about bacteria. From bacteria to the prokaryote to 10,000 genes. Then about first eukaryotic system, bacteria has around 4,000, 5,000 genes. If you pull all the bacteria in the tree, then first eukaryotic system 10,000 genes. Second transition is the from 10,000 to 30,000 genes. That is first vertebrate, they end up Cambrian, that 530 years, million years, that 10,000 to 30,000. This is the transitions. During these millions of years, what has happened? Duplications of the existing genes and some of acquiring new function of the duplicated genes. Some of them are gene families. Some of them are only one gene is functioning, other genes are pseudo genes, they are not active. So all this, the kind of the mechanism did work in this, right? So if you look at that, the duplication may involve single gene, part of the gene, or whole part of the chromosome, or the entire chromosome. The duplication of that can occur. So that's why the genomes have evolved in different organisms. The whole polyploidy after seen that. So this is one of the very well established and explained situation. One gene is present, duplicated. When you duplicate it, what is the selection operating on this gene? One of the situations here, the genes stay similar, the selection present on both genes are active. That's the multi-gene of the family. Out of this two genes, one will be degraded during the course of evolution, millions of years. Only one will be acting because that is beneficial, others are not beneficial. The third process here is that one gene functions similarly, other gene acquire new function. New variations are there, new functions. Remember, like hemoglobin in plants, hemoglobin in thin hemoglobin, there are different kinds of the embryos hemoglobin and adult hemoglobin. We have different polypeptides. All these have evolved as the complexity of the organism in millions of years. That is the example we can give. Yes. Lateral gene transfer. That is transformation. This terminology I hope my students know about transformation, transduction, and conjugation. So bacterial, any piece of DNA can transfer on this. This can happen in the eukaryotic system, where the eukaryotic system have nucleus, mitochondria, and chloroplast DNA. These can undergo lateral transfer. We do have many of examples of this. Nuclear gene or nuclear DNA moved into chloroplast, or chloroplast DNA moved into nucleus, or nuclear DNA moved into mitochondria, mitochondria DNA moved into the nucleus. We do have several examples. So lateral gene transfer is also one of the phenomena called variations in the eukaryotic system. And the variation based on the nucleotide variation, we can classify bacteria, eukarya, and archaebacteria based on the genomic variations. And this eukarya, we do have mouse and human, human and chimpanzee. We are 99% identical in genome. 6% of the gene are variant in the chimpanzee and human. That's why chimpanzee is different from the human. But 99% identical the genome we do see. So we evolve the language or the learning or the communication gene and so on. So to make the students aware of what is the success story in the genomic studies in our genome. Happiness gene. Have you heard of the happiness gene? We do have a gene coding for this 5-HTP, and which will regulate serotonin. Serotonin should be present. If the serotonin is regulated properly, we will be happy. If this is not regulated properly, even if they give 5 lakhs in the college institution as salary, we will not be happy. No, 5 lakhs is not sufficient. My son, I have to pay for my son fees or daughter fees, we say. Okay? Many times, even you have everything, you say, even we eat all the food. After eating the food, oh, this is not good, this is not good, she has not done this. Frankly, they do not have the right kind of regulation of serotonin. That's right. You have to enjoy that, to make regulation of the serotonin properly, then only we enjoy that. Otherwise, we have everything, we will not regulate. This is the epidemiology has been cloned, analyzed. If people have any kind of problem, they indirectly they activate the regulation of serotonin. When they undergo depression, doctors give this as a medicine. Sports gene. ACTN3 is the protein responsible for fast muscle fiber formations. If this is not there, even if you spend a lot of the money, exercise, everything on them to get the Olympic gold, it may not happen. And already Upendra gave another example in the morning. We need to know our genomic variation. Then don't tell the ladies that I don't have a cooking gene, that's why I'm not preparing. Every day take me out for the for the fast food. No. Okay? So yes, every character speeches have their relations, but it's left to us to how to make use of to understand this. Crime gene is also partly associated with monoamine oxidase gene. People have seen this low level of this oxidase product in the suicidal people when they taken off. Then this is the one cause the antisocial activity recorded. In some cases also they say that they look into that, then they'll also activate this or increase this production. By many means, by giving external, they can also monitor them to reduce their antisocial behavior. Genome editing, I'll stop. I'm not going to talk anything about it because Upendra is going to talk hours together. And the genome editing is excellent techniques, and we can use for any purpose now. And I think you're going to cover that. I'm not going to talk about it. So, the application of genomics now, genetic profiling, hybrid crop, lifestyles have social impact as man form, neo eugenics. So we can apply the genomics to create transgenic plants or animals or more production, genome editing to have a more production, wide application, keeping the ethics under control. Genomic revolution to create new jobs and improve economy. Government is not advertising any course, even I think 26 lecturer posts in zoology or botany or biology. They advertise for thousands of graduates who are there, how to accommodate them. So, a genomic revolution, new companies are coming up. They are hiring more than 50 students, and our PhD holders are working in MedGenome in Bangalore only, getting a good salary and that because of genomic revolution, genomic study. We do partly teach in our MSc course the understanding all this kind of radiation, understanding of the genome. Combined genomics help to understand all types of anthropological extinct organisms. I hope Upendra might tell something about the Nobel Prize this year. Someone thought we got the Nobel Prize looking back to the ancient where the Neanderthal and the Homo sapiens undergo process and they produce that also. Every evidence that we got it from. He made it bits and pieces, he collected, he amplified, he proved that got the Nobel Prize this time, that is the anthropological instinct organism can be traced. The very important tool is the genomic tool. He created new tools for understanding of this. Many new microbes found in gut for the human behavior. He also touched about the microbiome in our gut. That also tells about behavior. If your food was not good, I don't think if it has caused dysentery, the people would not have sit properly like this, right? So microbiome, whatever we have, also contribute for our activity. One of our studies recently published in a brain and health research, gut microbiome from the artisan samples shit we collected, isolated the DNA of the bacteria we classified, that also contributing for their artistic behavior and so on. That is one of the hot field now, gut microbiome research. So the analysis of genomic variants of COVID-19 also contributed to create a variety of vaccines, and some of the vaccines were working this now. And first human genome, the health samples, they can take now the genome study, any dead cell and dead tissue can take and process, and you can understand better about this. So genomic study is run the whole world. Another any system you take, another at least 50 years of your 50 or 70 years, because the longevity time yours is easily 100 years. Okay, because the kind of medicine we are going to have, the genomic medicine we are going to have, kind of the lifestyle you are going to change because you want to live, you want to enjoy life. So this is more important. So to conclude, genomics is genomic inspired thinking. Take up the challenges. When you ask me when you started understanding genomics, I was a postdoc in genomics lab in MIT, 1988. That was the time controversial human genome project. I used to spend a lot of my time reading in the immense library, very good articles I used to get. has started to, even now, Robert Smith's first article published about to start this, the Human Genome Project, I have my collection there. Okay? So that inspired thinking from that day, I'm talking about late 80s, even now, any new things, it's stimulating. I'm thinking now, I should have gone 20 or 30 years later than what I want, because a lot of fascinating things are happening now. We can do wonders with this kind of. Don't worry about the facility. You have to have mindset and hard work. Information helps the society in their use, eliminate new hypotheses, and understand help of our planets. They have application in every field, health, agriculture, fishery. I have not covered anything about agriculture because the scientists are coming tomorrow. They talk about agriculture. I have not covered anything about agriculture. Whatever I know in our field, I wanted to just put forth the examples here. And this is the change in the society towards the personalized, targeted genomic medicine. So, many of my MSc students, academic fellows, PhD scholars, collaborators like Upendra, funding agency, different funding agency, helped me to think and work and ask many of scientific questions. Thank you. I hope now you know a little bit about genome. I'm not making you to afraid of your genome, genomic variation. One of the doctors, when he went for the DST grant, the doctor, was the chairperson for the DST Health Science grant, they say that I wanted to do genomics of genetics and genomics of dyslexia, that already families have problems of the disease. Why you want to diagnose and tell them that the variation, that is a problem? I had a fight with him, but I did not get the grant. Because later I got another grant of that. So there are people giving negative effect of that. Let's not worried about it. Let's move positively. Think about, there are many negative aspects. Oh, if this kind of genomic we know, we can create the Cold War, we can create bio war, and so on. Yes. If you look at our mythology, Hindu mythology, our Ramayana, Kurukshetra, all Mahabharata, there was also problems. They are not free from this. Why do you worry too much about that? We have to have legislation and go. Good thinking. And we can enjoy. You have to love the subject, enjoy working, explore all the possibilities. That is the way. I'll stop it with now discussions. Yeah. There are several genes have been identified, about 18 genes globally. 5 new genes identified are Indian specific. That's one of them is the photogradity. Ultimately, what we suggest that in another paper we published that suggests that identification early, giving one-to-one counseling, one-to-one the communication skill, special school. Abdul Kalam was a dyslexia, and our Amitabh Bachchan's son, he is also Abhishek, he is also dyslexic. Doesn't mean that there's a problem. We have to identify, and some of the, and all will not have the defect in all the genes. Remember, breast cancer is, tomorrow I will touch a little bit about that. All will not have all the gene defect there. Whichever the gene is defect, 1 or 2 genes, you have to correct them also. So dyslexia, main problem is that identified, called as compensatory mechanism. That the neurons have plasticity. If this pathway is not blocked here, they can explore another pathway, they can communicate. Problem here is cell-cell communication, neuron-neuron communication there. So if this is blocked, if this pathway is blocked, the channel, another it will open. When it will open, give continuous training. Remember that the college people, continuous training, that will work. That is one of the management of that. The other, properly we can do have medicine, identify proper the gene, it can enhance their activity. Yeah, that's different. Yeah, it's different. That's also have a genetic factor, but we have not studied that. Any other question from this side? Yeah.
Namaste, sir. Is it true that vitiligo, tonkaile, is repetitive for the seventh generation, or is it just a misconception?
Two things here. As you already said, every disease, even infectious diseases, with my way of putting that, do have genetic component. Doesn't mean that only genetic component or genetic factors working there. Environment also important. So if environment induces, then it increases. Again, environment, lifestyle. The kind of food what they take also, those metabolites, those kind of things can also influence that. It inherits there. Sometimes again, the many factors are involved. Genetics is again we say chance factor. We can identify that what is the factor and which factor will go to the next generation difficult. Only after knowing that the factor, genetic factor in the next generation, we can identify it runs in the family. If it is recessive, sometimes it will eliminate it as homozygous. In heterozygous condition, it takes more time to eliminate. In a Darwinian selection, if recessive cannot be eliminated in the particular generation, it takes several generations to eliminate. If it is dominant, if that dominant mutation is that, if that is not there, it will not affect the next population. It depends on what kind of mutation. Again, one more phenomenon is that penetrance and expressivity. I already discussed in the morning the polydactyly. The gene is mutated, but it's not expressed completely or penetrated completely. Half of the finger is named. Okay? It again depends on, so all genes are not completely expressed and penetrated. Whereas the tall plant or dark plant, there the complete expressions are there. So this is another phenomenon and we have to consider for any kind of complex diseases. It's not just simply saying genetics is not good. What is that going to happen there? How much is penetrating and expressing is also important. Any questions? On this floor? Sure.
Thank you, sir, for giving me an opportunity. I'm an infant in genetics. And at the outset, I want to congratulate you and thank you on behalf of all the participants here for catering the post-lunch academic festive lunch, sir. That's what I want to tell. And in addition to her question, in our family, when dyslexic child is there, yeah, we had a lot of learning disabilities, but identifying his disabilities, when we train, he is having amazing memory. Like, is it a unique feature?
Yeah. There's certain processing of they will have problem with the mathematical process, but they are creative. They are very creative there. So we have to identify wherever they are good. Some of them are not able to write properly. I mean, in this dyslexic, let's not worry about that. Creative art, they can do that. So we have to identify their interest and train them, and they are not any inferior to any other individual. They can manage them also.
Do the males have more genes?
Do
to the males in human population, as you shown in one of these slides, each chromosome is having so many genes like that. X chromosome is having, we have homologous chromosomes, and you have non-homologous. I mean,
Right.
and additional genes you have also. So, do the males have more genes than females?
Cannot be, because Y chromosome has only 300 genes. If you compare the two X chromosome of 1100, so then we'll have 800 less gene, but not the number of genes they're affecting here. What kind of genes are expressing and working, that's important. Okay, because X chromosome more genes, and in homologous chromosome they're there, in Y chromosome they're less.
So, one more question, if you don't mind. He said, we have 90, I mean, there are 8 billion people, none of us are similar, we are all unique. And though we have 99.6% of genome the same, only 0.4 varies. How is that we are unique? Because we have the coding and non-coding sequences, do they change, interchange in each person as and when it's required, as per our necessities? What is that?
Yeah. Okay. Good. I got your point. Two things I can say. When this sperm has been produced after meiosis, in the one meiosis, 4 sperms have been produced. Each one is unique at that particular meiosis, one meiosis. The next meiosis process, they may not be same. So every sperm is a unique, not recombination. One person have recombination A, one sperm, another has a B, the third one may not have that in that particular section recombination. That's why every event is a unique. Similarly, with the egg also. That's one about the recombination, also introduce variation. The second thing is that our environment. We do have the same kind of thing. If the inducing environment you have, if somebody started scolding you, somebody started appreciating you, depends on which kind of the gene, your own gene. If, for example, here when the ladies are sitting there, if you bring a bunch of cockroaches, if you throw on them, some of them are there handling them, they will not respond. Others will run away, others will change. No, immediately the sensitivity, resistance, you can easily test that. So similarly, environment for every individual, their environment plays major. As if sometimes the environment, the culture, whatever the culture inculcated is not genetic, is non-genetic inheritance also is there. From morning, some family get up at 5 o'clock, 6 o'clock, some of them get 9 o'clock, and late rhythm, they changes. Just as if that they have the genetic inheritance. No. It's again our culture, our the habits, and how we maintain.
When coding and non-coding sequences are designated, they never get changed. And apart from the mutation. Okay.
Yeah.
Sir,
Yes.
one more question.
They might change again transposon. Any other event if that is they can change. Otherwise, there is no change.
Sir, current gene.
And again, one. When you say that, this only I'm talking about the genomic, the germ line. In somatic cells, they undergo inductions of the cancer, and they will amplify. They will change. The cancers, tumors, any other tumor cell. Somatic cells it can undergo the changes.
As far as the crime gene is concerned, all of us have crime gene, number one, and due to the environment, that's what we say, and if that gene is active, do we be like a Dandapalya gang, and I think you would have seen that movie also, and you would have sequenced, because people were telling at that point of time, ಗಂಡು ಪಲ್ಯದ ಮೆಂಟಾಲಿಟಿ ಹೇಗಿದೆ ಅಂತಂದ್ರೆ ಅವರು ಕೊಲೆ ಗಡಿದ್ರು ಅವ್ರ ಮೆಂಟಾಲಿಟಿ ಆಗಿದೆ ಅಂತಂದ್ರೆ ಈಸ್ ಇಟ್ ಬಿಕಾಸ್ ಆಫ್ ದ ಎನ್ವಿರಾನ್ಮೆಂಟ್
Yeah, the first one is that first answer. Everybody has the same gene, okay, and the expression level varies there. So expression level varies depending on the environment and the product. So they always they think that, for example, if you believe Swami Vivekananda, he says that 100 times things, the good thing will do the good things only. 100 times we think that, no, Hadimaga. Right? One day invariably our we Indians will do that. Right? So it depends on the environment where you.
Inculcation of the culture of
Yes. Yes.
practice. Right, sir.
Any other question? This side, one question. Somebody should ask this. You ask. Again, if you have any questions, we can ask after we open the start or tomorrow morning after my talk also. Still the things are open. We already posed 2 questions. Upendra is going to pose some more questions now after his talk, and let's have challenging the interaction.
Thank you for the excellent session, sir. It's the time to break for tea now. We have a tea break of 15 minutes.
Please, let me just walk it off. ನನಗೆ ಇದು ಮೈಕ್ರೋಫೋನ್ ಚೆನ್ನಾಗಿದೆ ವೈಲ್ಡ್ ಕೊಟ್ಬಿಡಿ ವೈಲ್ಡ್ ಕೊಟ್ಬಿಟ್ಟು ಹಾಂ ಪ್ರಯಾಣಿಕರು ಏನು ಕೊಟ್ಟಿರ್ತಾರೆ
Because the left hand is a very red.
this one this one
Something like we do have the cheap left hand, right hand, more of left and right hand side. Also the TBox, TBX cheese. That's why I'm not concentrating on the genetics. 20% genetics, 50% of environment for our daily life. I'm not here to propagate only genetics. Environment also very important. Upendra, she is coming with answer. Some answer.
That the homologous recombination that takes place in
What was the question?
all and
No.
Mutation ಆಗಿರೋದು ಸಾಧ್ಯ. ಸರಿ. Spastic children, gene function vary ಆಗುತ್ತೆ.
Generally, very safe wording exams ಎಲ್ಲ ಇದಾವೆ ಆಗತ್ತೆ ಬೇರೆ hopping in the variation.
Any questions in because I botanist and biologist. Just based on any questions.
Same tandem repeat sequence. The replication is ಹೋಗ್ತಾ ಇರ್ತದೆ ಮಿಸ್ ಎರಡನೇ ರಿಪೀಟ್ ಹೋದ್ರೆ ಮೂರನೇ ರಿಪೀಟ್ ಹೋಗಿ ಕ್ರಾಸಿಂಗ್ ಯೂಶಲ್ ಆಗಿ ಎರಡನೇ ರಿಪೀಟ್ ಅಷ್ಟು ಬಡಿ ಫಾಸ್ಟ್ ವೇರಿಯೇಷನ್ ಜಾಸ್ತಿ ಅದಕ್ಕೆ ಕೆಲವೊಂದು ಈಗ DNF ಪ್ರೊಫೈಲ್ ಬೇರೆ ಏನಿದೆ ನಿಮ್ಮ ಪೇರೆಂಟ್ಸ್ ಬೇರೆ ಏನಿದೆ because ಅಲ್ಲಿ ಬಂದು ರಿಕಮೆಂಡ್ ಯಾವ ಬಂದಿತ್ತು ಗೊತ್ತಿಲ್ಲ ನಿಮ್ಮ ಮಕ್ಕಳನ್ನು ಬೇರೆ ಇದೆ.
Sir, not for the genes, not for the genes. They do not play any role in the physiology of normal. Sir, morphology or physiology of normal.
ಮುಂಚಿತವಾಗಿ ಏನು ಹೇಳ್ತಿದ್ರು ಜಂಕ್ ಹೇಳ್ತಿದ್ರು.
That's what he studied, and we are not updated. That's why I said I'm an infant in genetics.
Micro RNA, small, it's non-coding. Micro RNA into ಹೋದ್ರೆ everything 6,000 6,400 micro RNA genes ಕೊಟ್ಟಿದ್ದಾರೆ.
Apart from 20,000 to 35,000, what we have secrets.
non-coding like RNA, RNA.
Oh, not coding. You gene posted 22,
25,000 coding. Even a non-coding. Non-coding, ಎರಡು ತರ ಜೀನ್ ಇದಾವೆ. ಕೋಡಿಂಗ್ ಜೀನ್, non-coding. ಕೋಡಿಂಗ್ ಪ್ರೋಟೀನ್ and other tRNA ಕೋಡ್ ಆಗಲ್ಲ. RNA ಕೋಡ್ ಆಗಲ್ಲ. Yeah, but they are called non-coding genes.
They get the stability to the.
Wait, no, they involve in the process of the translation. So, again, microRNA, 1988, on the first time plants are loading microRNA. Gene silencing or shooting. I mean, now they are identifying almost all of the breast cancer, regulation of the genes. Their specific microRNA. Okay, 6,000 microRNA. They are ಅವ್ರನ್ನೇ ಟಾರ್ಗೆಟ್ ಮಾಡ್ತಾರೆ, ಅವ್ರು ಬೇರೆ ಜೀನ್ ಟಾರ್ಗೆಟ್ ಮಾಡಿ ಬೇರೆ ಜೀನ್ ಟಾರ್ಗೆಟ್ ಮಾಡಿಲ್ಲ. ಈ ಫಂಕ್ಷನ್ಸ್
born late become
ಅಷ್ಟೊಳಗಡೆ
ಮುಂಚೆ ಇನ್ಸ್ಟಿಟ್ಯೂಟ್ ಆಗಬೇಕಾಗಿತ್ತು ನಾನು after hearing your lecture I should have
Most of the genetic disorders we highly respect is that the formulation of those treatments or products is expensive, or is that the things used are completely expensive.
no, produce one monoclonal antibody or one injection for 1 month of breast cancer.
So that is 16
Yeah, one monoclonal antibody for HER2 gene for breast cancer gene. For every month they have to pay. To produce that 2 ml or 5 ml, how much sacrifice they have to do, how much the experiment they have to do, the blood they have to do. Yes. In rabbit they create, or cell line they create. They have to grow so many of that, so many reagents. Yes. The whole process is expensive.
lots of expensive rather than things with
Process, but it's coming down. So, that's what we said that human genome project, one nucleotide, $1 they spent, $3 billion spent. Yeah, if you have one and a half lakhs, if you have, I can get the sequence complete yours and do an analysis. What kind of defect you have? Maybe people have to predict that what probable gene at what age you might get activated. Yes. Family clinical data is targeted studies, targeted genes. Family, you have some diabetes, and we look for all the 300 genes and all that. It's possible. One exome say 25,000 gene, madam. Madam, now first assume 75,000 spend ಮಾಡ್ತಿದ್ದೆ. hundreds ಸಾವಿರ ನಿಮ್ಗೆ ಜೀನ್ ನಿಮ್ಗೆ 20 ಜೀನ್ ಏನು ಕೆಲಸ ಮಾಡ್ತಾರೆ, ಹೇಗೆ ಕೆಲಸ ಮಾಡೋದು ಬೇಕಾ ಕಳಿಸ್ತೀನಿ. ಬ್ಲಡ್ ಸ್ಯಾಂಪಲ್ ತಗೊಂಡು ನಿಮ್ಗೆ ಅನಲೈಸ್ ಮಾಡಿ ನಿಮಗೆ ಒಂದು DVD ಇದೆ, ಅದು ರಿಪೋರ್ಟ್ ಕೊಡ್ತಾರೆ. 100 ಸಾವಿರ ಎಷ್ಟು ಬಿಗ್ಗೆ ಮಾಡ್ಬೋದು, ರಿಪೋರ್ಟ್ ಪ್ರೊಫೈಲ್ 2 ಸರಿ ಆರ್ಡರ್ ಮಾಡಿಸ್ಬಿಟ್ರೆ ನೀವು ಬಂದು ಹೋಗುತ್ತೆ. Again depends on what kind of
dyslexia ಆಗಲ್ಲ ಮಾಡಲ್ಲ cerebral
indications. For this man, we have this kind of this man. Then only they will do it. Otherwise, they will not.
ಯಾಕಂದ್ರೆ ನಮ್ಮ ಪಕ್ಕದ ಫ್ಯಾಮಿಲಿ ಸೆರೆಬ್ರಲ್ ಪಾಲಿಸಿ ಇರೋದು 15 ವರ್ಷ ಸ್ವಾಧೀನ ಇಲ್ಲ ಸ್ವಾಧೀನ ಇಲ್ಲ ಮಾತಾಡಕ್ಕೆ ಬರಲ್ಲ ಇಷ್ಟು
ಅಲ್ಲಿ ನೋಡಲ್ಲ ಸ್ಯಾನಿಟರಿ ನೋಡೋದು ಲಿಮಿಟೆಡ್ ಇದೆ ಅಷ್ಟೇ. ಅವ್ರು ನೋಡೋದು ಒಂದು ಮೇಜರ್ ಸಿಂಡ್ರೋಮ್ಸ್, ಡೌನ್ ಸಿಂಡ್ರೋಮು,
ಸೆರೆಬ್ರಲ್ ಪಾಲ್ಸಿ ಗೊತ್ತಾ?
ಅಷ್ಟು ಅದಕ್ಕೆ ಮಾಡ್ತಿರ್ಲಿಲ್ಲ. ಈಗ 4 ಮಾತ್ರ ಇದ್ದಾರೆ ಇವುಗಳಷ್ಟೇ. ಸೀರಮ್ ಪ್ರೋಟೀನ್ಸ್ ಇದಾಗಿದ್ರೆ ಡೌನ್ ಸಿಂಡ್ರೋಮ್ no cost, full expensive. Because now that they have some problems, indications and sensitivity analyze, then they have to go. It's expensive, but they have to go. And even the couple should know all these things before going for that. If we have this kind of where to go. As I said, if you rare disease, government will support. So support medicines, get the medicine, diagnosis.
Even it is there in percentages, for some it is very
Last
severe, for some it is
CV,
85%.
It depends on what is the effective.
A friend of mine
Dyslexic
had see around 85%, so she used to get busy and she used to but now she is yeah. But now she's taking pills like from 3 years. They had told to do the examination.
Yeah, he has lots of interest.
Explore. Neuron growth to
As per the necessity, as per the requirement.
So which will define because understanding
funding
right? Yeah. Now you will have. I know the kind of where the students want to learn. I have no problem. There is a big place called internet. There is everything about me, my email, everything is there except for my personal mobile number. So you just write it.
you have to validate it.
You talk to him, then if he says that the use will be taken, then we will take it.
It's easy to convince him than you ask the questions. As a learned student, now internet is there. You can select any persons, any places. You can identify the area. Right? There are hundreds of people, like Upendra. Then somebody will respond to you. Then look into that. If you are interested, you can respond. This is it. If yes, you
So I will tell you a story. There is for the alcohol drinking, there is a enzyme called alcohol dehydrogenase. Okay, if you have a good alcohol dehydrogenase, even if you drink, you will not get a kick, right? So one this Australian was drinking on the beach, right? So somebody said, that, "Why are you drinking?" Because I just removed my alcohol dehydrogenase, so I'm celebrating. Right? You're going to talk. So many of these things, even if there is a defective things, if you have a environment and surrounding, it will be fine. And the many of the like the intelligence, whatever sir said it is not one, it is multifactorial. You will have many more. And the other thing, remember that should I give them hint? more the one question. Okay, anyway, they can catch it. It is fine.
They are
Yeah. So there are something called say I'll give you an example. We have a family, 6 people, 5 had epilepsy, same mutation. The sixth one also had the same mutation, right? But the sixth one was fine,
They started giving the clue. If you are
right?
here,
Six,
you can
six,
get
six person was completely fine, right? So that you can say that how they have this mutation, but how it is able
ಪೆನ್ ಡ್ರೈವ್ ನಾನು ತಗೊಂಡಿದ್ದೇನೆ ಬೆಳಿಗ್ಗೆಗೆ ಓಕೆ ಸರ್ ನಿಮ್ಮ ಹತ್ರ ಇದೆ
So if
ಇದೆ
you have
ಓಕೆ
mutation, doesn't mean
ಮಾರ್ನಿಂಗ್
there are rare conditions, but it doesn't mean that you will have the people.
ಹೇಗಿದೆ ರೆಸ್ಪಾನ್ಸ್
Okay, there are something called as the modifier. So modifier is another, is a complex, another mutation, is another part of the gene, another part of the chromosome, or say some other thing. It will enhance or suppress the phenotype, right? If it's in mutation, which will enhance a phenotype, it is called as the enhancer modifier. This one will suppress is called as the suppressor, right? These are the second sign, right? Now somebody can answer the whole thing, but yeah. Yeah, please. You don't have to introduce me. Okay. Yeah.
Welcome back. I request all the students who are standing outside to come and sit in your places. Today's last session, that is session 4, is going to be started now.
Okay, they will be very busy, so we will wait for them. While waiting, I just want to activate your neuron again, right? Yeah. So with a few more. Can you just put it down a little bit? So, yeah. So so these are the things which you have been taught in the class all the time, and I'm going to ask you, okay? And you need to tell me logically what you think about. So I will, can you just use it? Otherwise, I don't need a mic. So okay, you have heard Darwin's natural selection, right? If you are a biologist, if you don't know Darwin's natural selection, then you should not be taking biology. Okay? Right? From 11th standard, you study natural selection, right? So somebody tell me in the natural selection, we always talk about Darwinian fitness. What is the Darwinian fitness? What defines the Darwinian fitness? Huh? Oh, I don't need it. I just need the mic. Yeah, okay. Otherwise, I can stop.
Oh, this is.
Okay. So who will define me what is Darwinian fitness? Yes.
If two organisms are in the
No,
same
no organism. What is Darwinian fitness? I just want one.
genital variations come with it.
You see, right? He is out of IAC interview. In one question out. Right. Yeah, tell me.
Then if any one of you are not interested, don't disturb the others. You can leave the house. We want you to listen whatever the discussion is.
See, why I'm asking you this question is that as a biologist, you need to know it. And believe me, once I explain you, then you will not forget it for life. Right? Yeah? So what is the natural selection? Darwinian natural selection, we talk about who is selecting what. Okay, Darwinian fitness, I will define you in one sentence, is nothing but the individual reproductive fitness. Okay? When you talk about the Darwinian fitness, right? Fit means not silly muscle building or something, right? Fit means when individual is able to reproduce. That's also fertile individual. If you are in fertile individual, you are producing it, then Darwinian fitness is 0. Even if you have thousands of sons and daughters, I'm talking about the insects, they can have in millions. But if they are infertile, then their Darwinian fitness is 0. at the individual level, Darwinian fitness can be zero. But at the population level, even if you are infertile, your Darwinian fitness will never be zero because your genes will be having in your brothers, sisters, or somebody. If you take care of somebody and you adopt somebody and that person is producing children, then you are increasing the fitness. Okay, in the human context, that is why Darwinian fitness at individual level it can be zero, but at the social or population level it can never be zero. Is that clear? Yeah, you will never forget, right? Yeah. So, okay, we understood Darwinian fitness. Now, what is natural selection? Darwinian natural selection. Nature selects. Right. Who is selecting? She says that the nature selects. Even including the teachers, please. When I explain, don't think that I'm making fun of you guys, right? Okay, even the teachers, because I really want to make you understand the concept. Because you see, I have been teaching teachers from 2012 in the teacher training program in Salakere for IAC second campus, where we do teacher training. And believe me, so far, except for 1% of the teachers, nobody can answer this natural selection. They teach, but they have never answered it right. And as a biologist, I think you should know natural selection. All right? Yeah? And so in natural selection, when you talk about it, who is selecting what? So she said, your name? Uma said that it is nature select, and your NCRT books in the 11, 12 standard say environment select, right? Uh, so I'll give you the answer because there is not much time. But remember, natural selection name was given just to be different from artificial selection. Okay, in the artificial selection, people have been doing artificial selection and breeding for 10, 20,000 years, 10,000. At least we know that in the maize or in the dog breeding, people have been doing artificial selection. In the artificial selection, what you do is that you pick the desired character, right? If you want bigger character or say golden retriever, Sir was talking about it, so you choose the dog, right? You breed and you find a character which is a long hair and look like this, you select it and you just breed that with a similar kind of thing. So that what you are doing is that is segregating the genes with that particular phenotype. Okay, that is the artificial selection. So in artificial selection, the person who is selecting, right? If it is environment, right? Yeah, it cannot select. Environment change every time, every second. Now also environment is changing for you guys. Okay, right? And it changes for everyone, not for one person. Unlike artificial selection, where the selector is selecting a specific trait, right? In the natural selection, what happens is that the environment changes for everybody. So if we hit this room to say 65, only people those who have lived in the extreme summer and things, they will withstand. Other will by 45, 46 follow. Right? Okay. So the individual who can withstand 65 degrees for say few minutes, it means that the individual has variation, what Sir was talking about, some genetic variation. Okay, right? To withstand the heat. Okay, that is why they are able to survive. Yeah. So in natural selection, your NCRT book is wrong when it says that the environment selects. Okay, right? Environment doesn't select. The individual who have got a variation to withstand or increase fitness, those, in other words, the one that he said adaptation, who can adapt to the changing environment, right? So it is the inherent property of the individual, okay? They get selected.
In other words, if I am environment, I'm not going to select. Right? I ask the same question to everyone. The person who answered, that person, because of his or her intelligence, make herself or himself selected by it. Right? So, unlike artificial selections, right, in natural selection, individual make himself, herself, or itself selected. Right? Okay? There is a very thin line to understand it. Okay? Right.
Uh, because in the discussions I wanted to put that. There are two ways to distinguish, as I said, thin line, Darwin way of natural selection, neo-Darwin way of natural selection. Darwin said if you read his the book on the Dobzhansky, origin of species of Dobzhansky, 8 lines he described what is natural selection. To put it in one line, Darwin said, fitted one will survive, not fitted one eliminate. Okay? Whereas neo-Darwinian, after considering the genotype or genetic basis of selection, they say that the genotype which has the differential survival and reproduction. Differential survival, if the two individuals are there, the two genotypes, the one which has the differential survival and reproduction, that will be selected, that individual will be selected, that population will be selected compared to other one. So now the genotype, genetics is added there. The genotype, for example, your scoring marks. If you take the scoring mark, somehow this lady or this boy has whatever the kind of question, they write very nicely. You have been selected differentially. You are selected among 100 members or 20 members. That trait only, the selection doesn't mean that you are good in everything there. Similarly, the natural selection is the differential survival and reproduction, that's a near Darwinian way. Darwin, whatever the artificial selection is said, that is the one picked up by near Darwinians. So they put it because Darwin time not much genetics was known. Even though Mendel contemporarily said that the knowledge was not there. Okay? To make it so, whatever the opinion said, genotype is very important. And all this genotype put into this environment, this particular environment, somebody can grasp proper faster than the other one. That is the differential. That is the selection. A particular trait. But if questions, whatever Upendra says, 5 sentences, somebody can repeat all the sentence. Somebody may not be able to repeat that also. That depends on your persistency and your genotypic. I will not take much time. So,
Okay.
So you have to remember these kind of things there.
Right.
Many textbooks, you don't read only one textbook, compare it with other textbooks, sometimes misconceptions will be.
Not only misconception, your interpretation is also very important, right? So, in one way to just check your interpretation, I will ask you one question, very simple one, not difficult one, right? Which organ tissue you think in your body is the most important one? Okay, let me ask this way: if you have to choose whether which organ tissue you want to be in your body, which one you will? Huh? Well, come on, this is a very simple question, right? And let's answer it. Brain, how many brain? Hands up. Only few are, still have more than 80% are not decided. Come on. And the other 80%, what you want to be? Why? Big brain, I would say that they will say that brain controls everything, right? Yeah? Okay. And the heart? It pumps blood through and the love is about the heart. Right? Yeah. Right. Okay. Yeah. Love. Okay. Good. Anything? Every organ is important, right? Um, but personally, if you ask this question, every tissue organ is important. But you should be able to articulate that how and why you choose a specific organ, right? Okay, I'll give you. Okay, today, if you have problem with the heart, you can replace heart. Still, you remain as your identity. You won't lose your identity. Okay? That correct? Yeah. So in that way, you remove your hand, even your lungs. Okay. Now there are artificial lungs which can be put. In few years time, still you will remain. In other words, I replace myself, even my face and everything by plastic surgery. Inside, I will remain as a Upendra. Right? Is it? It is because my brain says it. Right? So if you retain your whole body, and you now, let's say we have Elon Musk Neuralink, okay? He's making a Neuralink which will define your, you can put a chip in your brain and you will start behaving like a Google. And you will know everything. Okay? In those conditions, even if you look like a, say, I may look like a Upendra, but my identity will be lost. Okay? Yeah? Because now I will behave like any one of us. Those who transplant, Neuralink, all of us will start thinking similar. And Elon Musk can control also. Some kind of, right? Okay? Yeah? So what I'm trying to say is that when you change your brain, you don't remain your identity. That is why brain is the most important. Because identity, brain defined. Now, this is, you agree with me? Brain is important, right? Now, if I tell you that the microbe present in your gut actually defines how your brain develops, then which one is more important? Which one is more important? Microbe, right? Because microbe can define how your brain develops in the mother's womb. Okay? Very nice paper by actually one of our students who was working in Australia and Singapore. Just published cell paper last year in November. Okay, so the microbe defines how well your brain develops because the brain, if they stop receiving things, they stop thinking, then your identity you will not be able to. Right? So during COVID 2020 December, I went into coma, 30 minutes dead, right? Completely blank, I'm telling you. There was no, in the 30 minutes, no brother, sister, wife, son, daughter, nobody. No relation. I died, right? After 30 minutes, I woke up. Of course, then I saw somebody telling that your time is not out, so go back. That is how I come back, right? Okay? Yeah? I mean, if I would have not stayed Samundi here, Mysore, it would have been somebody else. Okay? But that's the God, if you believe in God. I'm telling you my truth. But then I realized that every day we die. When you are asleep, when you are in deep sleep, your identity is not there. No prime minister, no president, nothing. Okay? Yeah, that is why one of my students asked, we were talking about the whole longevity, we should make longevity, blah, blah, blah. He said that, sir, already India is so populated, right? Why you want to increase longevity? Yeah? Why you want to cure diseases? Right? We are talking about disease, curing disease, this thing and this. If you don't produce any kids, then it is okay. But when you have a kid, or you have a mother, or you have a father, you want them to be healthy and die. Okay? You don't want them to suffer. When you have a kid, you don't want them to suffer rolling on the. That is why we do research. That is the thing that drives me to work. It is not a question of curing or not curing disease or like increased longevity. It is about that when we are alive, we should do something so that the most important thing, lesson you take it from home today is that we should work in such a way that our next generation should have a better future than the one we have. That's all. Right? If our children, our future, if they are not going to have a better future, then there is no point in we doing workshop and studying and this whole thing. So think about it, right? Okay. In that direction, if you have a circle, all genes we know is variation this thing, right? Where is the technology? And believe me, this Nobel Prize winning work, right, is there already. People need to discover it, that's all. 1980 onward, people knew this enzyme, Cas9 is there, thing. But you think about it is that it took from 1980 almost 3-4 decades for people to realize that it can be. It similarly, Indian Ayurveda system, Sarakha said that you have the gut and your liver properly, and you will never suffer from any disease. You know Sarakha, father of Ayurveda, right? Now, only 2012, when Western science started following that actually micro can control behavior, micro can control things. 2012, it's not even a decade. But Indians knew it for last 4,000 years. But we never follow it scientifically. The way what we call as the rigor at which you do science, okay, it was not there. We believe it, that's all. Whether somebody follow or not, it doesn't matter. But the Western countries, they have been asking questions. What if you have this micro, if you have this metabolism, if you have this compound produced by the micro metabolites, how they can induce the character, all these things. Now we know that the microbe can control our body. Microbe can control our brain. Tomorrow, if some people from those who work on the gut microbiome system, if they get the Nobel Prize, we will say that we knew it from 4,000 years. Just like it happened with the circadian, right? Circadian clock, we said that, oh, we knew it for last 2,000, 3,000 years. Okay? So, what I'm trying to say is that you need to pull up your socks and be different. You all are different already. But somewhere you need to pull up your socks and say that I want to be this. That's it. Okay? And that cannot happen overnight. You have to work. You have to organize yourself. And this is the take-home message. After that, if you have defective genes where you can't understand anything, how to cure it? How to change it? Okay? Some of you would be very old to change it. Okay? As we talk about, say, epilepsy or neurodegenerative disease, it's very difficult because once all the brain which contains all the knowledge and everything, memory, if those are dead, there is no point. Okay? That happens in the Alzheimer and Parkinson's diseases, right? Okay? But believe me, today Alzheimer disease can be predicted with 86% accuracy at least 30 years before by simple PCR. All right? 17 years before, Alzheimer can be predicted with 94% accuracy. So it means that you can take precautionary action, right? Okay? Yeah, but how many of us will be able to take that test? Whether government will insist and whether you will be able to reduce the money, because at the moment you need to take the CSF, which can be done by very trained doctors, right? Tomorrow, if we can do it from saliva, if we can do it, because the Alzheimer starts at your gut, in your vagus neurons, the neurons which innervate your blood. And the Alzheimer, those precursors form into your gut. And this was work done by one of our Mysore products, my very good friend, a Mysore Maga, Kiran Bhaskar. Okay? You look at the Kiran Bhaskar. I haven't spoken about him, but he is in Cincinnati Children's Hospital in America, where he is one of the only few people who got tenureship from the first day. Because in America, to get tenureship, you have to really work hard. But for him, he got tenure from the first day. I don't want to talk to you about Kiran, but you think is that the Kiran was a, I mean, Kiran had third class mark in his second PU. He could not speak English, he could not speak Hindi or anything when he did degree with us. Degree with me in St. Philip. Okay, that person, right, today is world's renowned neurobiologist. Okay, so I'm not telling that you should become, because only Kiran is born only once and he will be only once. Okay, right? And you are just like Sarah was telling, Thomas Alva Edison, he was only one person without any degree, without any schooling. He made more than 300 patents. Okay, so everybody can be unique. And the common factor about all these people is that they never stop working hard. The moment Kiran came to know that by working hard one can become smart and think, we were friends, reading together, working hard together. Okay? So you choose your friend circle also very wisely. Right? Yeah? Okay. With that, if you have a still defective genes, how we can remove it, right? Or how we can tune it, right? This will happen really in few years' time. Everything can be corrected. Okay? So this is it. So that what is that? We can use the most one thing is the CRISPR. So that you will have change in the as we talk about changes in the sequence. You come down to the some gene. You say that there is a defect. How you will do it, right? I told you Marfan and all these things in the morning. So these are the most common mutation identified with genes identified which are associated with the phenotype. Okay, if you have defect in some of this gene, they will lead to this phenotype. Okay, now how you can correct it? So if you look at the human gene database, this was done around 4 or 5 years back, but you have the access. There are 3 databases for the mutation. Okay, so one is the human gene mutation database, where all the mutation which you get it from your parents, inherited one, right, is listed. Yeah, human gene database. Then you have human somatic mutation database, which happen mostly because of these cancer and things, right? Okay, when you are alive in the somatic mutation database, right? Okay, those are there. Then the third one is the human mitochondrial mutation database, where mitochondrial mutations are listed. Now, if you look at this mutation, you see by say 2017, I think you 2017 here. So by 2017, you already had people have reported 204,000 mutations which are directly linked to some disease. Okay, out of these 2004, only around say 10%, 5% actually, around 10,000 have the functionally annotated. Functionally annotated means that this gene mutation actually is responsible for the phenotype. That is where I told you model organism is very important. Now, so far, many of the genetic disorder, if you have it, right, it cannot be treated except for the few like the cleft cane. You people know that surgery can be done. Then you have metabolic disorder like inborn error of metabolism. I call it, okay, like the phenylketonuria, right? Those can be done by enzyme therapy. Or now today we have spinal muscular atrophy patient can be corrected. Then now we have hemophilia B which can be corrected, and I told you that costs a lot. Then the sickle cell anemia already data is there from the phase 3 clinical trial, where the sickle cell anemia have been cured now by using CRISPR-Cas. Okay, so all these things can be done. But the first one which was corrected was the adenosine diamidase, ADA, which we also work, okay, because this is a enzyme which can change A to inositol. A to I is a RNA editing enzyme. Okay, right? I don't know how many you know it, but don't worry. This is the enzyme actually which is now involved in almost every disease. And we have recently published a paper through Drosophila. We identified the gene which regulates the expression of the ADA or the adenosine deaminase is called as the ADA. Okay, it's called as the TEXI. I will talk about that TEXI. Now, see, when you have a mutation or defect in the every DNA replication is happening, there is a defect. The nature has its own method to correct it. So we have two kind of correction. One is called as the non-homologous end joining and the homologous end joining. In the non-homologous end joining, the defect is not, you don't use the one template. You don't use it. Okay? In the homologous, you use a template to connect it. Here, there is an additional base pair adding and you connect it. So the enzyme for this is different, enzymes for this are different. Okay? Remember, these are the something which you have in your body. Right? And it is working through and through. That is why mistake you have every cell cycle, cell division happen is 1 in say 10 to the power 6 in human. Okay, very rare. That is why we are all look like normal. Saras, you know what is normal, right? Now, how you will define normal? Sarah asked the same question, but I was not happy with the answer which you people give. In biology, what is normal? Huh? How you define normal? She says that if person is emotionally normal, that is right. So believe me, once you, I should not be saying it. Once you get married, now after the 6 months, you will all fighting with your husband, so we will not be emotionally stable, right? Okay, yeah. It means that all of us were abnormal, huh? Sorry, this example is not a good example, I think. Right? All right? Yeah? So that is not the correct. I mean, all of you are in the age where you want to have boyfriend and girlfriend, right? Do you really never fight? Believe me, you should fight. Because fight is the only way to understand others. If somebody says, a married couple says that we never fight, then that married couple is lying. Okay? If somebody says that you never fight with anybody, then those persons are lying. Okay? If somebody says that I am 100% honest, that person is lying. Okay? So never believe in this. Right? Yeah? There are a few exceptional cases, right? That is what we are calling as a exceptional, rare condition, right? So I have given you a hint now how you will define normal. Uniformity and proportion. You tell me in biological terms how much is normal and how much is abnormal. Okay, let me ask you a simple question now. Sir, I am going to, I will not take too much time here. This I will explain you in 30 minutes, but you need to learn here. What is null hypothesis? Somebody explain me what is null hypothesis. That's why you need to know maths. Because all our biological assumptions are based on math. Okay? So what is null hypothesis? Somebody very fast. Null. N-U-L-L. Null. Yeah. Null is zero, yes. So what is null hypothesis could be? Come on, yeah. Then I'm telling you null means zero. Come on, guys. Null hypothesis means that, that the variation should be zero. when you do any experiment, right, or anything, when you observe, 100% it should fit. There should not be any variation. That is what we call as there will be no variation. That is what we call as null, right? Null variation. But in biology, you can't have that. It can be possible in maths and theory and things, but not in biology. So the biology, up to 5% a mistake is acceptable. Okay? If you repeat one experiment 100 times and then 95 times, if you are able to reproduce, then your it falls within your null hypothesis. So the 5% is acceptable. The moment you can't repeat your experiment 95 times, it is 94 and 93, right? Then it is not acceptable. But again, in biology, it depends on the what experiment you are doing. Because in behavioral science, up to 10% variation is acceptable. Okay? Are you guys with me? It means that I need to give you lots of knowledge and your foundation. These are the foundation. Okay? These are foundation. Right? Okay? So null hypothesis. That is why if you look at the Mendelian ratios, still we call as the Mendelian paradox. Mendelian paradox because all the 7 experiments, contrasting experiment, monohybrid, dihybrid, he did it. All the things came within the expected value. Expected value means that if you without doing experiment because he was very good in right. So everything came within that. That is why we call it as it is too good to be true. But because we don't want to blame the father of genetics, so we call it as the Mendel's paradox. Okay? Right? Yeah? Is that clear? So normal is nothing. Normal depends on what you are asking. So it could be emotional, it could be high, it could be it depends on what is your question. If you are looking at the eye size, of course I am abnormal. My eyes are smaller than yours. But if any one of you come to Manipur, then you become abnormal. Okay? Yeah? Right? So you survey all the population on the what character you are looking at. Okay? For the running, for example, running 100 meters, all of us boys should be able to complete in 12 to 15 seconds. The girls should be in 16 to 20 seconds. But if somebody is running very fast, right, in the 9 or 10, those minority, the 5%, right, if you count it in the 100%, 5%, those exceptional ones are what we call other different. They are the abnormal. Those who can run very fast. And those who put it in the 95%, they are the normal. Is that clear? So how you define normal? Tomorrow, next time, if anybody asks that what is normal, you will be able to answer, right? Still, some of you haven't understood what I was telling, right? Because you have never seen a Gaussian distribution. Have you seen any Gaussian distribution graph like this? Yeah? If you look at the height, two short people, then those who are average, then those who are very tall, right? If you are looking at the tall, then the short and those who are medium, the last 5% who are really tall, they are the abnormal. Is that clear? Example? Done. Now, so these are the natural conditions. They automatically work. Now, how you can correct things? You think about it. Enzymes are already there in your body. It means that if you have a system where you can generate defect in the cell willingly in a specific genes of your target, right? Do you understand? In normal conditions, the cells will correct only when there is a defect, right? Okay. Now, forcefully, by willingly, if you generate a mutation, right, in the specific gene that you want to manipulate, then this enzyme will go and correct it, right? That's it. The whole genetic system on this hypothesis. Now, how to target our interest gene? Now, that is the question. That's all. Because you want to make a seizure, there is already a person in this, who will just swing machine, right? Machine. They will sew it. If you cut it, my point is that there is already a person who will like tailor and keep on sewing, right? Okay. Now the question is that where, how, in which gene you want to generate the cut. Okay, right? So that is what people did it. For a long time, people work by using homologous recombination. So you are providing if you want to create a mutation, you provide for the mutation. Okay? So the normal defective should be replaced by the abnormal, or vice versa. If there is already abnormal, you provide a template with a normal to cure it, right? And the efficiency was 1 in like 10,000, right? And the maximum efficiency they could bring was 1 in 3,000, right? So it was very, very rare. In 1980s and 60s, every mouse made by the homologous recombination was a cell paper or nature paper. Okay, because it was very difficult to generate such mutation, right? Okay, and then people started something called the zinc finger nuclease talents. Okay, so what happened is that we know that how you can cut and correct it. So if you have a now a enzyme which can cut the DNA, right, of your choice, then so people started using this, but each one these are artificially synthesized. Each one costs 3.5 lakhs. How many labs in the world will be able to afford that, right? 3.5 lakhs. And again, success rate was very, very low. So if you have a technology, as I told you, so that you can just put it inside and cut it wherever the gene, right? What you want is only the targeted gene, right? And this is how recombination can happen. And I told you that you need to have this construct to take this enzyme to the target gene. And the people knew that this enzyme from 1989, actually 1981. That was the first Indian postdoc who became PI later on in the MIT. Actually, he identified this enzyme. Okay. But he never did anything. It's just like in the autophagy. Autophagy was, discovered in 1963. After 1963, I don't know, people have been working on the red and things, even in India also. Nobody discovered it. Till 1993, remember? 1963 to 1993, for 30 years, nobody discovered it. And one Japanese guy, he, by mistake, didn't put the foot in his culture, he discovered it. And he became Nobel Prize winner. Right? Okay? Sometimes mistake, but the observation is very important, remember? Right? Okay? So, this CRISPR, right, stand for the cluster regulatory interface short palindromic repeats. In other words, repeats here. These are the DNA sequences. That's all. People identify it in the E. coli. Right? It is a natural system of immune for the bacteria. Because bacteria are infected by virus, right? Lambda 4 virus, right? Okay. Bacteria is just like an innate immune mechanism of the bacteria. Every time a new lambda bacteriophage infect the bacteria, when the lambda virus genome gets integrated, right? The bacteria genome retains some part of the lambda bacteriophage. So the next time the same bacteriophage goes, the bacteria will produce your RNAi thing, right? Okay, a sequence which will go bind to this bacteriophage sequence and kill it, digest it. So it is a bacteria's own immune system. Okay, right? Okay, so and for the doing this, bacteria has all this repeat with the bacteria sequences. There is a protein called as the Cas9. Okay, you need enzyme, isn't it? So you have the palindromic sequences to read it and to activate it and cut it, right? You need a sequence enzyme. These are called as the Cas9. Amongst all the Cas9, smallest is the Cas9 here. So that is why we use this Cas9, because Cas9 doesn't require all other domains. So the Cas9 is the smallest. Now we have many artificial Cas9 Cas enzyme up to Cas13. So the Cas13 actually can bind to RNA unlike DNA. Okay, so this is unique. In other words, tomorrow we should be able to even edit RNA sequence. Okay, so this Cas9 was used. You can see that. So the first paper that this how this CRISPR and the Cas9 could be involved as an immune mechanism for the bacteria. Actually, this Elena Sororia is from Poland, and she would not publish this paper. She thought that she has identified new things. And you think about it, that she could not publish this paper. She sent to Nature Science, thinking that it is a immune system for the bacteria, and she was rejected left, right. And finally, she published actually in an unknown journal, almost like that time it was a very new journal. You can see the microbiology. Okay, 2005 microbiology. So sometimes it is not necessary that only the big journals they publish only the new thing. If somebody who is referee, they don't know the work, they can't figure out the novelty of the things, then it can happen. The same thing happened with you asked him, right? Okay, when the people said for the Down syndrome, he didn't narrate the story, but I can narrate you. Is the Down syndrome, the grandmother's age has an influence on the, whether how many the Down syndrome will have it or not. It doesn't matter on the mother's age. Usually the old mother age will produce more Down syndrome, okay, risks. But what serves novelty was that it doesn't matter on the mother's age. The grandmother's age is important. That time we didn't use, sir didn't use, even though I insisted that he should use the epigenetic, but he didn't use it. If he would have used the epigenetic, then the people would have been talking about him as a pioneer in epigenetic, because epigenetic came only in 2007 and 9. Okay. Today, if you look at that paper, which is one of the most cited paper for sir, all the epigenetic people working in the field cite this paper. I think this is the most cited paper, right? Yeah, right. Okay. And the worst thing is that people, those who sit in the granting agency in India, they didn't understand that work, and sir didn't get any grant for that. And only when the Americans say that this is a novel work and thing, that is where people started recognizing. So that happened in the Indian history. Ramanandan to everyone. Only when they went out, Western people recognized and they got the recognition in India. So in India, we will never recognize anybody bright people. Like me, I will never be recognized, right, sir? I'm just joking, right? Okay. But what I'm trying to say is that in India, we like to just pull down. We never support anybody. Okay? Just live that attitude, believe it. If you can tomorrow, so that you'll be fine, right? In the same way, CRISPR-Cas, okay, these people were like thrown out. That you are talking nonsense, you don't know what you are talking about, right? Yeah. So, but afterwards, She kept on working. Her students actually went to postdoc in the Western country. They started working. They identified that actually these are the things. And the one of the first paper what they published was in the I think science. So they published that actually it is the immune system for the bacteria, right? Okay. The once it is established that the CRISPR-Cas system is the immune system for the bacteria, whether what is the mechanism? That is where this lady who got the Nobel Prize, right? Can you read her name? Yeah. Huh? Carpenter, whatever, right? So she showed that you need a sequence. Beside this thing, you need a trans sequence in it, all right, to bind exactly. So the identification of this sequence was very important because these CRISPR will not work if you don't have this trans acting sequence. That was important. The other thing is that at the same time, this lady who was working, these are the something transacting and how it works. At the same time, in the well, I'm forgetting her name. My COVID-4. I just can't remember people's name. So if you tell me your name after some time, I will forget. Only person I remember maybe Ramchandra sir, Rangnath sir, or my wife's name. Okay. um, what was her name for? Huh? No, Carpenter was the other lady. Jennifer, Jennifer, she worked on the RNA binding protein structure. So she was working on RNA binding protein for a long time. Okay, actually she worked on the splicing structure of the splicing mechanism. But then she went to the thing and she got a call one day and she started looking. Oh, we should look at this DNA binding. It has a potential. That's what I mean. You know, suddenly changing the field, this is what we call as a visionary. You should be able to decide that this is exactly what I should do. She was working on the RNA binding protein for a long time. She had good papers, good lab and things. Suddenly somebody said that, okay, I saw this poster in Europe meeting. Somebody was talking about this Cas9 protein, which can bind, take the RNA to specific region of the DNA. This would be fantastic, right? Then she started working. She deciphered the Cas, all this structure because she had the facility, right? Because you need to know the mechanism, right? So she looked on these things. These all are science paper, okay? She identified. Then one day, actually, both of them came, okay? Carpenter came in this meeting and said, okay, I'm coming to this meeting in Brazil. Can we meet? Okay. So this is what we call as the networking. In science, you need to have a good networking. If you can't do something, you should be talking to people, right? Sir knows my networking. Where I went for my postdoc, Professor John Sparrow, actually, I got to three people, John Sparrow, Jim Wegros, and Sandy Bernstein. Three of them I have got paper, okay? We have paper. Three of them I wrote to send me mutation that I am working in the, Massey University. I don't know anything. We don't have anything. Can you provide flight? And you think about it, all the three people sent us flight. But among all the three, a person who wrote me long email that wish you all the experiment, blah, blah. You think about it, that time there was no email. I'm talking about the facts. He sent me facts with the protocol, how to do it. I still have the Western protocol, 25 pages protocol. He sent me how to prepare protein, and I kept it. I used to go to, one fake machine person in the market right here, and I used to go and collect it. Then that is where I decided that I want to join this guy as a postdoc, because he had that, time and thinking about a person that what I need, I don't need anything. And take it from him, I have met all three of them, but Professor John Sparrow used to come to IAC every alternate year after joining IAC, because he we develop this fatherly sonly, connections. Okay, so you can have it. So this is what I'm talking about, networking. Networking is important. So in this context, these two ladies left the conference. They started walking. I was told that they walked almost like 10 kilometers. They were so much into the experiment that they forgot how much they have walked. Okay, in Brazil. That is where they developed that, okay, we need to come your protein, whatever you have expressed for your structure, we need to bring it, and we need to have all my reagent for the RNA, DNA, and put it and see if they can edit anything, right? So this is the structure. By having a structure, doesn't mean that it will work, okay? You need to do experiment. This is what they discussed in that meeting. After some time, they started doing the experiment. Let's not worry about it. Where it can cut, okay? They design it and they knew it, right? Okay. So this is what how the Cas9 works. So you need a guide RNA to take the Cas9 protein to the target DNA. What you need? A guide RNA to take the Cas9 to target DNA. Okay. Because Cas9 has got all the activity for the cutting DNA and opening it, helicase activity and everything. Okay, so you need to have that, and a local system we call it the palm sequence. You need it to click the guide RNA and Cas9 enzyme to the DNA. You need a click lock system, and that sequence is called the PAM sequence. Okay, photoactivator sequence. Okay, yeah, it can be only 3 nucleotides. So you need that, and you just put it inside, and they will do all the experiment. Okay, this is the postdocs actually, those who did the one from Carpenter's and one from Jennifer's lab, Jennifer Dortner. Okay, so they did the experiment. They did thousands of this experiment actually. That is the whole thing. These two people, they didn't get the Nobel Prize. The mentors, those who decided to do the experiment, they got the Nobel Prize. But these two came on doing all the experiments. You can see it was not working for many, few months. It was not working until they decided to put the trans-acting elements, something which they were thinking that the enzyme should be able to work without that small sequence which Carpenter discovered. But it works only when those trans-acting sequence was put. That's it. But you can see it. It can cut. Okay? For that, they got the Nobel Prize. That's it. Okay? Actually, another person should have got the Nobel Prize. But then, in the last few years, we have been talking about the women empowerment, women empowerment. So they didn't want to include a guy for this Nobel Prize. The person who actually used this technology, let's not worry, these are the different Cas9. Now you can modify the Cas9 in such a way that you can edit, because the Cas9, if you put it with for example, Adar, I told you, deaminase, then it will be able to edit A to I. In other words, if you have a mutation in the A, it can be changed to C. Okay? Right? So the Cas9 has been modified to edit even single nucleotides. Okay? So I will have it. So that this Cas9, you have only 3-4 things. Single RNA, then you have the target palm sequence, you need it, then you have the enzyme and inject it, then you generate defective things, and if you provide homologous sequence from outside, then you can correct the sequence. If you don't have a homologous sequence, you can generate many indels. In that way, you can generate mutations, right? And if you have the Cas9, which has the ability to correct the single nucleotide, then you can correct this place at the single nucleotide level. That's it. Okay? So these are the things what you needed. So I told you this is a locking system, all right? Okay? So these are the 3 nucleotides you can see. Now, it started almost in 1987, 89. I told you the first enzyme discovery till here. Now, thousands of CRISPR-Cas based medicine is just waiting for just to come from clinical trial to the market. And it, unlike other adenovirus based and thing, it will be much cheaper. Okay, right? Yeah. So the person who should have got the Nobel Prize is this guy, Feng Zhang. Okay, and you should read his what we call story also. It's very interesting. Never giving up. And most of the Chinese, of course, they don't give up, right? And I do. I also belong to that race. I never give up, right? Yeah, all the Northeast people actually they migrated from China. Don't means we have that lineage. Sir told you now most of the Chinese people they migrated, right? And one of the migration those reached the northeast part of the say Thailand, Burma, Manipur, and Mizoram. I mean, I belong to that, right? I also don't give up. And this guy even worked in the he never had any job. He worked as a marketing person. It's very interesting. I didn't know about him. So I started reading about it because he needed money for his family. So he acted as a clerk, right, in one of the companies, and he acted. He worked as a photocopier at that time, yeah, early 1985. He worked as a photocopier because he had to run his family. But he was so much interested in the research, then he started working in the company, as a field agent. Then he started working as a lab agent. And most of these things actually he did it. He knew it already. Only when he started publishing from the company, then the Broad and MIT they offered him a position. They're also known as the assistant or associate professor. Remember, unlike in India, MIT and Broad, the tenureship is only 40%. It means that the 60% you will join. If you don't deliver, go out. Right? Okay. It's so much, right? Quality. So he was given in the MIT, and that is where he saw for the first time that CRISPR-Cas can be used to edit specific gene in cell culture system, in the vertebrate cell culture system. So he should have actually, he's the person who showed that the CRISPR-Cas can be used to edit gene in real. Okay, and I mean when the announcement came, I was just waiting that I knew that he was also shortlisted, and I was expecting that these three people will get the Nobel Prize. But next day I found that it's women empowerment, only the two women got it, right? Okay, nothing against women. I'm just telling you that how people can discriminate. Okay, and I support women, by the way, right? We have more women faculty in my department than any other department, right? You look at it, MRBG department, right? Okay, we recruit. So, but what I'm trying to say is that this guy deserved to be the Nobel Prize. Yeah. But luckily for him, CRISPR-Cas9 in America, the patent went to him, not to the other people. Okay, right? The patent how to use editing CRISPR-Cas, actually the patent belongs to him, not to Carpentier or Jennifer. Okay, whereas in Europe, all the three of them have got a patent for international patent. Okay? Patent matters a lot because for the at least for the next 7-8 years, still you think about it, 2005, 2012 only they were able to show it. 2015 we saw it, right? So we still have almost like 12 years. If anybody wants to use, they need to give money to these people. So at least, even though he hasn't got the Nobel Prize, he will die with being a billionaire. Yeah? Okay? So that's it actually. So he was the first person to show gene editing in the thing. Today, you can precisely cut, do things, and there are close to 25 companies working on it. And they, at the moment, if you look at the clinical trial registry, there are close to 1,500 CRISPR-Cas based medicines which can be used for the human, and these are all undergoing human clinical trial. Okay, right? And some of them, like as I told you, for the sickle cell anemia, clinical trial phase 3 is over, and it worked. If it has worked in the clinical phase 3, means that it will take only a few months to come into the market. Okay, right? So the problem still is that if you want to edit, one girl asked now, if you have a dyslexia gene, how you will correct it, right? So Sark is very nice, which is currently available thing. Tomorrow you should be able to do cell therapy, what we call it, right? Cell therapy means that you see most of these particles, you want them to go inside the cell, right? So at the moment you have the lipoparticles and things to take it. The challenge for the next 15, 20 years is that how to deliver these things cell specifically, tissue specifically, and see, adenovirus didn't work so far was that you think about it, 1996, the adenovirus gene therapy, it worked on principle. One girl got cured from immunodeficiency. But it didn't come to the market is because when you inject even millions of virus, adenovirus, it doesn't survive in body. It gets thrown off in the body. Okay? Right? So how you can make this as a cell specific? So you should be able to have this carrier of the CRISPR-Cas. How you will inject it? CRISPR-Cas just can't inject and it should go and penetrate and correct all the genes and remove it. Which you see where you have seen such thing? Where you have seen these things? Where you just inject nanoparticle and then get cured? Where you have seen? Huh? Where? That is where you people even don't watch the Star Trek. Do you know Star Trek? Star Trek. Please watch Star Trek Voyager, for example. Okay? Star Trek Voyager started in 1995. All the medicine which we are talking about, nanomedicine, correction and everything was done in the Star Trek that TV series. It is available on Netflix. You please have a look. And it is really good if you want to become leader also, because the captain, how she conducts herself is amazing. I wish all the Indian leaders should watch that Star Trek. I know, I mean, they wouldn't have time, but nanomedicines, where you may take these things to deliver to specific tissues and organ and correction, right? Okay. Or otherwise, you take out the defective cell, correct it from outside. If it is a stem cell, you correct it from outside and then transplant it. Now, adenovirus people have developed is that people have developed adenovirus which are tissue specific. So, for example, recently, 3-4 months back, muscle specific adenovirus they have taken. Adenovirus could take only small sequences, around say up to 2 kb. Today, adenovirus 9, for example, can take few kb. Okay, and you can have 2 adenovirus taking big genes. For example, there is a adenovirus which have been made by our people in Bangalore, Narayan Hospital, where Arca is the PI. They have developed a system. It is an Indian one, adenovirus, where they put whole 2.4 megabytes into 2 halves. And they have put it, and these 2 adenovirus come together to form the full length. It's amazing science, actually. Okay, and it is done by our actually Indian scientists in Narayan. Okay, and it is coming up for the treatment of DMD in India, actually. Okay, so this is what the challenge will be: how to deliver it. All right. So this is at the moment liposome-based delivery. So you generate a liposome. We also work in this where we have with the professors, one professor from IACS, one chemist, right? So we developed this called as a cell penetrable peptides. Small peptides which can just take anything inside cell very fast. Okay, it's a patented again, US patent. He has Suraj Sarkar in his name. He is in the IACS, okay, Indian Cultivation of Science, IACS, right? In Kolkata. That is the institute, one of the oldest institute where Raman started his career. Right? Yeah. So that is where we are into. So we do it. So what you need is that this thing. Why it works is that this can be done by anybody in a small lab. You don't need a big facility like adenovirus and things. You need big facility. But for the CRISPR-Cas, you don't need a big facility. Even we do it every day in, day out in my lab. Of course, we don't use it for the human. We use it for the zebrafish and Drosophila. But we do it day in, day out in the lab, for example. Okay? So it just works in the script of, it just works. Okay? So these three people, as I told you, patent one, this guy won in America, these all three of them won in the Europe, and they will be earning in millions. And these are the way people have been using it. CRISPR and gene therapy, this is the stage where we have, I told you, 1,500, more than 1,500 clinical trials at the moment going on for the CRISPR vaccine. Many, even cancer, where there are multiple mutations. Okay, in cancer there are multiple mutations that arise in genes. Already one myeloma has been corrected by using CRISPR-Cas because you can target many genes in a same time to correct it. Not only one, but many genes. Okay, and they have done it in cancer recently, and some of these are listed here. I don't know if you can see it, but take it from me. These are taken from the clinical trial web page, and these are something which is undergoing clinical trial at the moment. Okay, so the medical application at the moment, it can be used for the cancer immunotherapy, tissue regeneration, gene therapy. Malaria has been successful. ICIP now has been successful, and a few insulin resistant thing in the diabetes and metabolism, which is in the clinical trial. Okay, so it can be used. So as I told you, the most important thing is that you find out whether it is a nanoparticle or the cell therapy, how you will be able to deliver it in the tissue or the cell specific way so that they go there and correct it. That is the major challenge, and you still have a time to contribute in this field. Okay, right? It works. So people have generated thousands of corrected them in the model organism. CRISPR-Cas works in the plant because the moment you remove the Cas9, whatever is modified in the plant, unlike other genetically modified plants, actually you can grow them without any problem. Okay, so the CRISPR-Cas9 for the rice is amazing. These have been generated. So the future is this thing. All right. So these are some of the companies which work extensively on the Cas9. And this is Eric Olson. Here you will see it with his name. Eric Olson is one very senior professor in America. So he has already shown that the CRISPR-Cas can be used to correct the mutation in dystrophy mice. Okay, mice which is suffering from DMD, Duchenne muscular dystrophy, was corrected. And similarly, you can see that at least the protein which is normal in the MDS, it comes here. So at 85, he has got a big company. So I don't know whether this company, if he will survive or not, it doesn't matter. But company will survive, right? Okay, yeah. So the good thing about the CRISPR-Cas, at least in the model organism like Drosophila, is that we have thousands of these CRISPR-Cas line which can be used. You don't have to make it. My point is that you just order it and you can use them. Then we do it. These are the different CRISPR-Cas9 lines which can be used. Whether you want to do, I don't know how many of you understand it. Different biology questions like say gene expression, proteins expression, all that you want to regulate it, you can use it CRISPR-Cas now, right? So I told you the TXC is a gene which we identify which can regulate ADAR or the adenosine deaminase. So we had a mutation and we found that mutation happened because of the insertion in this sequence. It is already published last year or this year. This year we published it, right? Okay. So you can see that the sequence insertion, you can see it. It should be here, but you have 2.5 DNA sequence insertion. And these insertions actually leads to a phenotype. Okay, the genes unable to express properly. And this is what we found. So to know that actually this insertion is causing the phenotype, we have to remove it. So we use the CRISPR-Cas9. You can see that we use the CRISPR-Cas9 to remove it. So we designed this. I don't know how many of you are with me, but what I'm trying to say is that you need to just design few primer, few sequence, you generate this thing, inject it in the fly line like this, and you can see that we have removed this, right? So this was big one, which was there, we have removed, right? And if you remove this sequence was responsible for the phenotype, you should be able to restore the phenotype, and that is what actually. So that this is abnormal, this is abnormal. Now, after removing the sequence, it becomes completely normal. Okay? So this is what something we do. We do it in the animal, but this same thing can be done on any tissue, any organ in the human. This is what we call as the proof of principle. Okay, so now what we have done is that we have now we are able to deliver this by using FDA approved nanoparticle called as the PCLA, so that we can have a tissue specific delivery. Okay, so we are doing all these things in using Cebrabis and Drosophila, right? Okay, so that you can see that the flight has been recovered. That's all, right? Okay, so I stop here, and if you have any questions, then I will answer it. Right? I think I should actually the 1 hour 10 minutes. Right? Yeah, any questions? Anything? I know it is late, but any questions? No? I'm around tomorrow morning, so you can ask me anytime. Get me during lunchtime. So I'm around. I'll come during lunchtime, not in the morning. Right? Yeah? Is that okay? Any questions? Anything? Anything which is bugging your brain at the moment? You know, two things can happen. You understood something, that is why you are keeping quiet, or other thing is the extreme case where you know you haven't understood anything, everything went here. Okay, right? And take it from me. This is exactly what I felt. You know, when I came back in 2004, I go with the IAC students. They must be very bright, talented. So I prepared my first lecture, very nicely with the whole new thing. And I personally taught them, right, this first class, right, developmental biology, very nicely, the teacher. Then, because I think they are bright, right? After the class, I said that, okay, give me a feedback, how about it? So they just, you know, IAC students, they just walk out, you know, right? They said, sir, good to see you. Then there is one student came back and said, sir, initially we thought that your talk is going to be low, your lecture is going to be talk because you are shouting, you are telling, and I said that, okay, but after a few slides, everything went just about, right? So it means that they haven't understood. So even for IAC nowadays, when I prepare slides, I make sure that I prepare just like any other place. Right? Yeah? But now, whatever I prepared is, I thought that it should be good for the any other place. But now that your foundation is not great, you haven't understood one thing. The other thing is that you know you have not been able to catch my program session, which is mandatory, right? This can happen in future. Yeah? No? You understood?
Yes.
Yeah. At least thank you that you understood.
Myositis, from which Samantha Prabhu is suffering from autoimmune disorder. Can it be corrected, rectified? At what stage that we shall develop? And what is the reason for that?
Myositis can happen because of the retention, because of the genetic, because of multiple things. Until we identify what is the root cause to be cancer. I wish I can say that every disease, everything can be cured. For example, to many of your eye disease tomorrow, even if you have the microspheropathia, which I talk about, only way is that you can't generate one another eye because you have only one eye, right? So only way you can do is the artificial eye, which is now already approved. FDA has approved the artificial eye. If your neurons are fine, actually you can put this artificial eye. So in a similar way, if it is a muscle autoimmune which is specific, remember autoimmune can be for antibody which can go for the whole tissue or the whole, I mean organs, multiple organs or the single tissue or there are tissue specific antibodies and it is because of the defect in the HeLa or anything, then it should be possible. But it is because of the expression of multiple things, then you need to target it. But as I said, sir, ma'am, muscle up to some extent you can cure because you have a stem cell population called the satellite cell where you can correct it. But there are many tissue in our body where you can't just do it because there are no regenerative ability they don't have. Like liver you can correct it, like blood you can correct, but many tissues where their regeneration ability is less, it will be difficult. Any other question? Any student? No? Right? You want to go home? Okay. Chalo. See you tomorrow.
If you have any question, tomorrow I can. Thank you for your patience hearing. Just for your
Thank you, sir. Thank you for the wonderful session. Now I would like to call upon stage our beloved principal madam and Professor Ramachandra sir to felicitate Professor Upendra.
Okay, you have to stay back to see me. But this can be done tomorrow. Not like this. I can go.
So we are at the end of the first day's session. Thank you all for your excellent patience. Tomorrow we'll continue with the sessions. Have a great day. Tomorrow's sessions will be starting from morning 9:30 a.m. I request all the such fellow students to assemble here before 9:30.
Okay.